Greetings to Professor Dr. Leslie. We are from Group 2 and our topic is about malaria. Malaria is a significant public health concern in many countries. It is an infectious disease that caused by Plasmodium parasite and transmitted through the bite of infected Anopheles mosquitoes. In our country, Malaysia, we experience the transmission of several species of Plasmodium, including Plasmodium falciparum and also Plasmodium vivax. Malaria is most prevalent in states with higher transmission rates, such as Sabah, Sarawak, and also certain areas in Pahang. This region have suitable environmental conditions for the breeding survival of malaria-carrying mosquitoes. Factors contributing to the transmission of malaria in Malaysia include the presence of human settlement in close proximity to forested area, which provide a habitat for Anopheles mosquitoes.
I'm going to explain about the biological pathway of malaria. First, when an infected female Anopheles mosquito bites a human, Plasmodium parasites in the form of sporozoites are introduced in the circulation and causes malaria. Sporozoites enter the human liver fast. Over the course of the next 7 to 10 days, the sporozoites proliferate asexually in the liver cells with no symptoms. In an animal model, the parasites, which take the form of merozoites, embark from the liver cells in vesicles, travel through the heart, and then make their way into the lungs, where they settle within the lung capillaries. The vesicles gradually fall apart, allowing the merozoites to go to the blood stage of the development. Following that, the merozoites penetrate red blood cells, erythrocytes, in the circulation and continue to proliferate inside of them until the cells explode. More erythrocytes are then invaded. Each time the parasites escape their control and enter blood cells, the cycle is repeated, resulting in fever. The cycle of asexual multiplication is broken by the certain infected blood cells. The merozoites in these cells transform into gametocytes, the parasite's sexual forms, which circulate in the bloodstream instead of multiplying. The gametocytes, which eventually grow into adult sex cells known as gametes, are consumed by the mosquito when it bites an infected person. The female gametes that have been fertilized grow into moving active ookinetes that pierce the midgut wall of the mosquito and mature into oocytes on the outside. Numerous thousands of sporozoites that are active grow within the oocyte. Eventually, the oocyte explodes, releasing sporozoites into the body cavity that go onto the salivary glands of the mosquito. When the mosquito bites another individual, the human infection cycle starts again.
Next, let us discuss an innovative solution in the fight against malaria: genetically modified mosquitoes. These mosquitoes have been altered using gene editing techniques such as CRISPR-Cas9 to reduce their ability to transmit the malaria parasite. Scientists can change these mosquitoes' immune response gene or parasite receptors via gene editing. This prevents disease transmission. Another approach is to reduce the mosquitoes' vector competence, making them less effective disease carriers. This can be achieved by altering the mosquito's immune system to combat the parasite or inhibit the parasite's ability to replicate within the mosquito. Population suppression aims to utilize GM mosquitoes to reduce population size. For example, introducing a gene that produces sterile offspring can decrease the overall mosquito population over time. One of the fascinating aspects of GM mosquitoes is the gene drive effect. Gene drive elements bias the inheritance of modified genes during reproduction, allowing the desired trait to spread rapidly within the mosquito population. Field trials aim to test the effectiveness of GM mosquitoes. These trials involve releasing GM mosquitoes into the wild and observing their impact on local mosquito population and malaria transmission rate. Now, let us discuss the benefits of GM mosquitoes. The combination of gene drive and CRISPR-Cas9 provides a targeted approach by modifying genes related to malaria transmission. By focusing on a mosquito vector rather than the human host, we can reduce the transmission rates without relying solely on human behavior or drug treatments. GM mosquitoes can specifically target mosquito population without affecting other organisms or causing harm to the ecosystem. The deployment of GM mosquitoes for malaria control requires regulatory approval and careful assessment of potential environmental and health risks. Regulatory frameworks must be in place to assess the safety, efficiency, and potential risks associated with combining gene drive and CRISPR-Cas9 technologies. Continuous monitoring and evaluation are required to track the changes in mosquito population, disease transmission, and any ecological effect.
Malaria is a serious health problem that affects millions of people worldwide. To prevent and control malaria, it is important to take a multi-sectoral approach that involves collaboration between different sectors such as health, education, and agriculture. The following are some recommendations for further research and action to prevent and control malaria. First, increase funding for malaria research. Second, promote the use of prevention methods. Third, address challenges in malaria control. In conclusion, malaria is a complex disease that requires a multi-sectoral approach for prevention and control. By increasing funding for research, promoting prevention methods, and also addressing challenges in malaria control, we can work towards eliminating malaria as a public health problem. Thank you.